There are moments in science when the closer you look, the more extraordinary the picture becomes. When the microscope reveals not simplicity but staggering complexity. When what appears ordinary from the outside is, on the inside, one of the most sophisticated biological events in the known universe. The development of a human being, like from a single fertilized cell to a fully formed infant capable of surviving outside the womb, is one of those moments. And the organ that makes this development possible, that mediates every breath of oxygen and every molecule of nutrition that passes between mother and child during the 9 months of gestation, is one of the most remarkable biological structures ever studied. An organ that grows from scratch performs functions no other organ in the body can replicate, and then, its mission complete, is delivered from the body and discarded. That organ is the placenta, and it is the unsung hero of human life. Today we are going to walk through the entire journey, from the moment of fertilization through the extraordinary process of placental development, through the weeks and months of fetal growth that the placenta makes possible, through the biological cascade that initiates labor and delivery, and through the final moments of birth. We are going to explain the science behind one of biology's greatest achievements, not in clinical language that distances us from the wonder of what is happening, but in the vivid, specific, evidence-based detail that reveals why every human birth is, in the deepest biological sense, a miracle. This video is for everyone who has ever wondered what really happens during those 9 months, for parents and grandparents, for people who want to understand the science of human beginnings, and for anyone who has ever held a newborn and felt the instinctive awareness that something extraordinary had just occurred, and wanted to understand exactly how extraordinary it truly was. Let us begin at the beginning. Fertilization is the event that initiates everything. It occurs when a single spermatozoon, one of the approximately 200 to 300 million released during reproduction, successfully penetrates the outer layers of a mature egg cell, the oocyte, in the fallopian tube. Of the millions that begin the journey, only a few hundred reach the fallopian tube, and of those, typically only one achieves fertilization. The competition is extraordinary. The attrition is almost total. When the successful sperm penetrates the zona pellucida, the protective glycoprotein shell surrounding the egg, it triggers an immediate and dramatic response. The egg cell undergoes a rapid change in its electrical charge that physically blocks all other sperm from entering, a phenomenon called the zona reaction. The cortical reaction follows within seconds. The egg releases enzymes that chemically harden the zona pellucida into an impenetrable barrier. In the span of moments, the egg transforms from an open door to a fortress, protecting the genetic union that is now taking place inside. The sperm and egg each contribute 23 chromosomes, half the normal complement of 46, to form the zygote, the first cell of a new human being, containing the complete 46-chromosome genetic blueprint that will guide every aspect of the person's development. Hair color, eye color, the architecture of the brain, the proportions of the skeleton, the specific configuration of the immune system, all of it encoded in the DNA of this single cell, smaller than the period at the end of this sentence. Within hours of fertilization, the zygote begins to divide. 2 cells become 4, 4 become 8, 8 become 16. This early cell cluster, called the morula, for its resemblance to a mulberry, travels slowly down the fallopian tube toward the uterus over approximately 3 to 4 days, dividing continuously as it moves. By the time it reaches the uterus, it has transformed into a hollow ball of approximately 100 cells called the blastocyst, a structure that already contains the fundamental differentiation between the cells that will become the embryo itself and the cells that will become the placenta and its supporting structures. This early differentiation, the separation of cells destined to become a human being from cells destined to become the organ that will sustain that human being, is one of the first and most consequential decisions in human development. And it occurs before implantation, before the pregnancy is even established in the uterus, in a free-floating ball of cells that is already organizing itself with a precision that is breathtaking to contemplate. The inner cell mass, the small cluster of cells on one side of the blastocyst, will become the embryo. The outer layer of cells surrounding the blastocyst, called the trophoblast, will become the placenta and the chorion, the outermost membrane surrounding the developing baby. Implantation occurs approximately 6 to 10 days after fertilization, when the blastocyst reaches the uterus and begins to burrow into the endometrium, the carefully prepared inner lining of the uterine wall. The endometrium has been primed by the hormones of the menstrual cycle, estrogen and progesterone, to be receptive, vascularized, and nutritionally rich, ready to receive the implanting embryo. The blastocyst attaches to this surface, and the trophoblast cells begin an invasion, extending into the endometrial tissue with a controlled aggressiveness that is one of the most sophisticated biological negotiations in nature. The trophoblast cells suppress the maternal immune response in the uterus, which would otherwise recognize the embryo as foreign tissue and attempt to reject it, since it carries paternal genetic material. They invade the maternal blood vessels in the uterine wall, remodeling them from narrow, high-resistance vessels into wide, low-resistance channels that will supply the massive blood flow the placenta requires. They secrete hormones and chemical signals that redirect maternal physiology toward supporting the pregnancy. The implanting embryo is not passive. It is a molecular diplomat of extraordinary sophistication, negotiating its own survival in the immunological territory of another person's body. The hormone that announces implantation to the maternal body is human chorionic gonadotropin, HCG, produced by the trophoblast cells from the moment implantation begins. HCG signals the corpus luteum, the remnant of the follicle from which the egg was released, to continue producing progesterone rather than degenerating as it would in a normal menstrual cycle. Progesterone maintains the endometrium, prevents the uterus from contracting, and supports the early pregnancy until the placenta itself can take over progesterone production, a transition that occurs at approximately 8 to 10 weeks of gestation, called the luteoplacental shift. HCG is also the hormone detected by home pregnancy tests. Its presence in maternal urine beginning approximately 10 to 14 days after fertilization is the biochemical signal that transforms the result window of a test strip from negative to positive, a moment that has changed millions of lives. The placenta develops rapidly during the first trimester, beginning as a thin layer of trophoblast cells and expanding into a complex, disc-shaped organ that, by the end of the third month, is already performing the functions that will sustain the developing baby throughout the rest of the pregnancy. Inside the placenta, the architecture is extraordinary. The fetal blood vessels of the placenta are organized into finger-like projections called chorionic villi that extend into pools of maternal blood, the intervillous spaces, that are continuously replenished by the remodeled spiral arteries of the uterine wall. Across the thin placental membrane, everything that the developing baby needs passes in one direction, and everything that the developing baby produces passes in the other. Oxygen from the maternal circulation diffuses across the placental membrane into the fetal capillaries. Carbon dioxide from the fetal circulation diffuses in the reverse direction for removal by the maternal lungs. Glucose, amino acids, fatty acids, vitamins, minerals, and immunoglobulins, the building blocks and protective agents of a developing human life, are transported across this membrane through a combination of passive diffusion, facilitated transport, and active transport mechanisms that together represent one of the most sophisticated biological exchange systems in nature. The umbilical cord, the lifeline connecting the placenta to the developing baby, contains 2 umbilical arteries and 1 umbilical vein, all surrounded by a protective gelatinous substance called Wharton's jelly and enclosed in a smooth, spiral-twisted casing. The 2 arteries carry oxygen-depleted blood from the fetus to the placenta. The single vein carries oxygen-rich, nutrient-laden blood from the placenta back to the fetus. At full term, the placenta delivers approximately 300 milliliters of blood per minute to the fetus, a flow rate that meets the extraordinary metabolic demands of rapid fetal brain development, skeletal growth, and organ maturation. The placenta is also the fetus's endocrine organ, a hormone-producing factory of remarkable productivity. In addition to HCG and progesterone, the placenta produces estrogen, human placental lactogen, which prepares the maternal breast tissue for milk production and adjusts maternal metabolism to prioritize glucose delivery to the fetus, placental growth factor, and dozens of other signaling molecules that coordinate the complex physiological adaptations of pregnancy throughout the maternal body. These adaptations are profound. The pregnant woman's blood volume increases by approximately 50%. Her cardiac output increases by 30 to 50%. Her respiratory rate increases to accommodate the increased oxygen demand of supporting 2 metabolizing organisms. Every major organ system in the maternal body is modified by the hormonal and mechanical demands of pregnancy, and the orchestra conductor of most of these modifications is the placenta. The development of the fetal brain deserves its own dedicated moment, because it is perhaps the most extraordinary aspect of the entire developmental story. The human brain begins as a flat sheet of cells, the neural plate, that forms at approximately 3 weeks of gestation. Over the following days, This flat sheet folds to form the neural tube, the structure that will become the entire central nervous system. Adequate folic acid in the maternal diet dramatically reduces the probability of neural tube defects, which is why folic acid supplementation before and during early pregnancy is among the most evidence-supported nutritional recommendations in reproductive medicine. The neurons of the brain, approximately 100 billion cells, are generated in a process called neurogenesis, most active between weeks 8 and 28 of gestation. At peak neurogenesis, approximately 250,000 new neurons are generated every minute. These neurons must then migrate to their final positions and form connections, synapses, with other neurons through synaptogenesis. By the time of birth, the fetal brain has already established the basic neural architecture that will support the infant's capacity for sensation, emotion, memory, and the early stages of social cognition. The brain with which a newborn enters the world is not a blank slate. It is a richly connected, experience-shaped organ that has been developing and responding to its environment for months before its first breath. By the end of the first trimester, week 12, the fetus has developed all of its major organ systems in rudimentary form. The heart began beating at approximately 3 to 4 weeks of gestation. The second trimester, weeks 13 through 26, is the period of rapid growth and refinement. The lungs begin producing surfactant, the critical substance that will allow the air sacs to expand after birth. The fetus begins to hear at approximately 18 to 20 weeks of gestation, responding to sounds from the outside world. At approximately 20 weeks, the mother typically feels fetal movement for the first time, a phenomenon called quickening. The third trimester, weeks 27 through 40, is the period of finalization. The brain undergoes its most dramatic growth. The characteristic folding of the cerebral cortex occurs primarily in the third trimester. The lungs mature sufficiently to sustain independent respiration. Maternal antibodies are transferred across the placental membrane, the gift of passive immunity that will protect the newborn during the first months of life. Before we continue to the cascade that initiates labor and delivery, here is a pause for you. What part of this biological story is most surprising to you? Have you had children or been close to someone through pregnancy and found yourself understanding something now that you could not have explained before? Drop a comment below right now. Your reflection matters to this community, and this is one of the stories that connects all of us at the most fundamental level of human experience. The initiation of labor is the outcome of a shift in the balance between the forces that maintain uterine quiescence throughout pregnancy and the forces that drive uterine contractility. As the fetus matures, its adrenal glands produce increasing amounts of cortisol, a fetal signal that travels to the placenta and shifts the estrogen to progesterone ratio, increasing estrogen relative to progesterone in the local uterine environment. This shift is critical because progesterone maintains uterine muscle quiescence while estrogen promotes contractility. As the ratio shifts, the uterine muscle becomes increasingly sensitive to oxytocin, the hormone that drives uterine contractions, as oxytocin receptor density in the uterine muscle increases dramatically in the days before labor. Prostaglandins, local hormone-like molecules produced by the uterine membranes and cervix, play a critical role in cervical ripening, the process by which the cervix softens, shortens, and dilates in preparation for the passage of the baby. A cervix that is closed, firm, and long at 38 weeks must become soft, short, and open, dilating from 0 to 10 centimeters to allow the baby to pass. Active labor unfolds in 3 stages. The first stage encompasses the entire period from the onset of regular contractions to complete cervical dilation of 10 centimeters, from early labor through active labor proper, in which contractions become regular, stronger, and more frequent, typically occurring every 3 to 5 minutes and lasting 45 to 60 seconds. The second stage of labor begins with complete cervical dilation and ends with the birth of the baby. During this stage, the baby descends through the pelvis in the precisely choreographed cardinal movements of labor: engagement, descent, flexion, internal rotation, extension, external rotation, and expulsion, navigating the geometry of the pelvic bones with mechanical precision. The third stage of labor, the delivery of the placenta, occurs in the minutes following the birth of the baby, as the uterus contracts powerfully and the placenta separates from the uterine wall. The hours immediately following birth are as biologically remarkable as the 9 months that preceded them. The first breath, taken within seconds of birth, draws air into the lungs for the first time. The surfactant produced by the fetal lungs in the final weeks of gestation is critical to this transition. maintaining open-air sacs that oxygen exchange requires. Within minutes of birth, the foramen oval between the two upper chambers of the heart closes as pressure changes reverse, redirecting blood flow through the now functioning lungs. The ductus arteriosus, the blood vessel that allowed blood to bypass the pulmonary circulation during fetal life, constricts within hours of birth in response to rising oxygen tension, eventually closing permanently. The transition from fetal to neonatal circulation, occurring over the first hours of life, is the most rapid and complete reorganization of an organ system that occurs at any point in human biology. Skin-to-skin contact between mother and newborn immediately after birth triggers a cascade of biological events. Oxytocin release in both mother and infant that promotes bonding and calms the newborn's stress responses, stabilization of the newborn's temperature, and the initiation of breastfeeding, which, if begun within the first hour after birth, delivers colostrum, the first milk, rich in maternal antibodies, to the newborn's developing immune system. The story that began with a single fertilized cell has arrived at its destination, not its final destination, that lies across a whole human lifetime ahead. But the first chapter has closed, and it is, by any measure, one of the most remarkable chapters in the natural world. The biology of human development, from fertilization to birth, is a story that has been unfolding in the same essential form for hundreds of thousands of years of human history. It unfolds in the same way in every human being on Earth, in every culture, in every language, across every variation of human experience. And at every stage of that story, the science reveals not simplicity, but layers upon layers of extraordinary complexity, molecular negotiations, hormonal orchestrations, structural transformations, and cellular decisions of staggering precision, that together produce the most ordinary and the most astonishing thing in the world, a new human life. If this video gave you a new appreciation for the biological miracle that every birth represents, whether you experienced it yourself, witnessed it in someone you love, or simply wanted to understand the science behind one of humanity's most universal experiences, please support this channel. Hit the like button right now, subscribe so you never miss a video like this, and leave a comment below. Tell us what part of this journey surprised you most, or what this video made you think of. We read every comment, and this is one of the conversations we are most grateful to be part of. Thank you for watching. We will see you in the next one.